And now we have Srishti, who's going to be the moderator for the panel discussion with Praveen and Mitali. So over to you, Srishti.
Thank you, Arushi. Thank you for the introduction, and thank you, Dr. Rahul and Dr. Mohit, for introducing the course and also, the importance of omics and the data integration part. So we look forward to the course. So we have eminent speakers and panelists with us today, Dr. Mitali and Praveen, and we would like to have them on board and discuss about the importance of multi-omics. So my first question would be for Dr. Mitali. So Dr. Mitali, if you could please briefly explain about omics and what do it refers to and its significance in the modern research nowadays.
Uh, so the omics is the study of the entire genetic, meaning the attributes of the entire genetic makeup of an individual. It can be any life form. It could be a virus like the COVID. It could be the human. So how they behave, how they are different, how do you know what is inherited? Any functional attribute which we see in an organism is an outcome of our genetic makeup, and how do we study them is all the technologies around it, and which is now become multi-omics technologies. So you start from DNA, it will be genomics, then you look at its expression, it becomes transcriptomics. At the RNA level, expression at the protein level becomes proteomics, lipid level becomes lipidomics, metabolite level becomes metabolomics. So you study the entire thing in entirety. So you need technologies. Earlier we would do one thing at a time. Now there are platform technologies where you can do all of it together. So that would be the aspect. And then all life forms, be it bacteria or be it human, we all have the same language, DNA, same AGCT. So as a result of which, same set of technologies can be used for all these diverse things. And there is also extensive sharing, so that you can understand meaning of one life form from studying the other life form. So it opens up a whole gamut of possibilities for understanding life on Earth. So as a result of which, if you study any from any perspective, you can apply it to different applications. So I think that's in a nutshell. I would like to say that this is all omics.
So thank you, Dr. Mitali, for briefly introducing about omics, its importance, and also shedding some light on the technical aspects of it. We would like to have pose another question for our panelist, Praveen. So we would like to know that, could you provide some insights on how the dynamics of genomics have influenced research in India over the past decade?
Okay. Thank you, Srishti. Thank you, India Biosciences. We are sitting with one of the pioneers in the genomics field. Professor Mitali is here. I still remember those days, ma'am. If you remember, 20 years back, you were working with IGVDB, trying to catalog and create the India's first, database for cataloging all the variations from across the different populations. And I still remember how complex as a project, and it's a multi-centered project with so many collaborators. I forgot how many authors were there in that paper, but I think something 150 plus,
200.
200.
200.
Imagine. So a massive project of that scale was undertaken about 20 years back to create. To start the initiation of genomics at a high scale. Of course, we've been doing genomics for many years before too. Professor S.K. Brahmachari is a pioneer in that area. But I think this particular project led to kind of looking at high throughputness or scale, or looking at large sample sizes and looking at large complex data to be cataloged in the country for the first time. From there till now, today we are talking about, recently DBT announced the completion of Genome India, and we've done 10,000 genomes now, whole genome sequencing. This is at a very high depth coverage of cataloging every single base pair of humans Indian population, covering about 70 plus population types or subtypes in the country. So I see that the way genomics has probably influenced is that whether it's human, whether it's plants, whether it's animals, whether it's bacteria, whether it's viruses. Um, it's greatly influenced the scale and the speed and in unraveling the complexities of different omics per se. I'll give you one example, a very common basic application during COVID. The way India took up, the sequencing project to basically look at creating, cataloging the variations of all the new upcoming strains, and I think that became a household, I would say, exercise where I think a common man understood the importance of genomics, the importance of sequencing, and today I think people understand what is variance. So that is, I think, so to me, I think it has come a long way, and I'm very excited to see where it is going. And as Dr. Mitali said, I think multi-omics is the way now, where people are looking at further complexities to combine, overlay data to see how we can understand biology even better and being more closer to the function per se.
Thank you, Praveen, for answering that question, because we understand, the importance of genomics and how we have come and progressed in the last 10 years, and now multi-omics is the future that we all need to look out for. Moving on to the next question, I would like to ask Dr. Mitali that, what are the challenges do we face in understanding the complexities of, the complex diseases, and also how the omics can help understand those complexities?
Complex diseases by nature are complex, right? It's not only genes which play a role, but it also is the environment, and how does the environment impact the expression of the genes, so that the same genetic makeup, maybe between related individuals, exhibit differences in the outcome. So you might have siblings having an equal chance of a predisposition, or maybe having a genetic makeup which is nearly similar, but they progress differently. And if you take this to the population, or to even different countries, then you will find that there is no set rules, it seems to be. And that's because the genome is very big. It's 3 billion bases, the human genome. And there are so many combinatorial possibilities because there is a huge amount of variation. So between two unrelated individuals, we would vary at a million places. Now, if you think that a fraction of these variations can actually impact the functioning of our genes, or overall functioning, then how many combinatorial possibilities can come out of it? And then how do you kind of have some kind of a prediction as to who are differently predisposed? That is what we want to know, because then we can actually prevent diseases in some people. Why do some people interact, have differences with respect to medicines or outcome, or what is the likely prognosis? So you want to keep on predicting from a person's genetic makeup, how likely is that person to be susceptible to disease, to have a adverse outcome if they have the disease, or how they could modulate the environment, and which environment they should modulate so that their genetic behavior is kept at optimum, right? So for all these things, you need multi-omic technologies, because you cannot, everything works in networks, and it could be the same network, but players might be different in different individuals, and so you need to study them in totality. So that's the reason that in complex diseases, multi-omic technologies have become like the bread and butter of trying to tackle it. So large number of success stories have come out. It's not that everything is known now, but we are starting to build these this knowledge up. And as the knowledge grows, and this is so high throughput, and people are making the data available, it also gives us an opportunity to do meta analysis and not do all the experiments ourselves, and port information. And that's where the computational part of the omics comes in, very, very important. So this is what our course caters to. There are advancements in the technologies. So if you just develop the skills, then you might be just limited to doing that much. But what is the foundational knowledge you should have? So as the technology increases or as the data increases, how do you tackle that and how you become relevant at all spaces is what this course is all about.
Yeah, I mean, we completely agree with you that, we can have multi-omics resolve so many complex diseases and have like a big, huge database which we can refer to, and also the importance of computational biology and bioinformatics. And the course that we all are offering helps us understand the importance and the relevance of, multi-omics. Praveen, you've mentioned about the importance and how pandemic has made us realize that the importance of genomics and multiomics. But then there's another question that I would like to ask you is that that currently what is the current status of the omics research in India, and where all we as compared to the global landscape where India stands?
Okay. um, so, I mean, my simple answer would be that we've done a lot, but there's a lot to be done. So I think with now the advances in technology available, we were talking about the whole advancements per se. So with the advent of technologies like NGS, and now new technologies like proximity extension assays, where people are trying to combine to study even protein or proteome at a large scale using the same genomics machines. With the availability of technologies, the way the world is adopting these technologies into large scale projects is at a different scale altogether. You look at UK Biobank, they've already done 500,000 sample profiling, for whole genome sequencing. And now, based on the data which is coming out, they've actually made certain guidelines in the NHS system that now every cancer patient or every rare disorder patient gets a whole genome sequencing in the clinical practice per se. So, and like this, there are zillion other projects. India too has done a lot of projects. We've had some, so IGVDB was one, but I mean, we have Genome India, we have Indigen, we have ICGC, we have ICGA, which is going on right now. We are looking at so many mycobacterium genome sequencing large scale projects which are being done right now. So, at every scale, the work has been going on. But I still believe, looking at the large population size of the country, we need to do more. And I think we need to do more multi-omic studies now. At small scale, a lot of labs are now beginning to realize to combine this layering of omic data and not just looking at one dimension per se. So as Dr. Mitali and Rahul explained that how people were earlier looking at either whole genome sequencing and maybe at the most transcriptome level. But now, if you can overlay with looking at large number of proteins together, there are about 2 lakh proteins, say in human side. So normally, when you're studying maybe 30, 40, 50 proteins together, instead of that, if you're looking at studying about 5,000, 6,000, 10,000 proteins together at one go, and if technology is available, that is where I think those adoptions have to start in the country. Some labs are doing it, but I think it's still at a very low scale. I would love to see more of that happening.
Yeah, I mean, we all of us here in the panelists and also the audience would agree that, there's a lot of scope of growing the multiomics domain. We have small labs doing experiments, but there's a lot of scope of expanding the multiomics domain and having a lot of sample inputs as well. Talking about the another challenge that currently we are facing is that, the clinical applications of multiomics and how we can, help and face or solve those challenges that we are currently facing in terms of the omics research and their applications in the clinical settings. Dr. Mitali, if you could answer that.
So, I think, like Praveen mentioned, our contribution in the genomic space is comparatively very, very low. As Meaning, if we think about our population size, our diversity, the number of patient pool which we have more participated in international projects by giving our samples to outside for analysis than really doing that much work in India in a collaborative setup. Genome India was the first biggest one, and also the ICGA. So there is an enormous scope, and I think what we need to do is we need to build an ecosystem of every stakeholder, and amongst them, the clinician become one very, very core important component, because the clinicians have a better understanding of a disease, and they have an handle to the phenotypes. But the genomic people in the labs have more knowledge about maybe what happens to the if to ascribe functionality. But if we do not make both of them crosstalk, then the right kind of an outcome what a clinician might at the end need for an application to be easily translatable into a clinical space might not be so easily done. So as a result of which, it now we need to have at every space hybrid people, doctors who know a little bit of genomics to have a good conversation with genomics people, genomic people to have a conversation with clinicians to be able to tune their analysis to much better way, computational people to be able to have the ability to talk with the doctors for an appropriate problem to develop their skill sets. So this is all very, very complementary need and complementary expertise which we need to gain. So what we have in in this diploma program, or whatever we have developed, it's in that philosophy we have developed this, that if we have this thing imparted to the students, they could have variable trajectory. It could be clinician coming into it and having a knowledge so that they can also plan better experiments. If they plan better experiment, then the service providers or the company people like Praveen will be able to get better reagents across to them, which are very, very useful. The genomics will also grow because the moment clinicians have started to acknowledge that this is the need, they'll start analyzing it. So it's a very needed area. Or at this point of time, what is most limiting is doctors think they have the samples, they can give it to a service provider does a routine analysis. Populations are different, cases are different, India is very different from Western world, all the standards are based on European population. The same diet is not there, the same environment is not there, the same exposures are not there. So we need to really, there's a major need for all these annotations. So in the clinical space, there's a huge amount of need and a huge amount of opportunity to integrate omics, and it's not only for the clinicians, but it is also for people who are going to do genomics.
Thank you, Dr. Mitali, for for making us understand that it is important for us to bridge the gap between the government as well as the private sector, so that we have stakeholders and a strong built ecosystem. Which brings to me, which brings to the last question that I would like to ask Praveen is that. Since already Dr. Mitali has explained about having an ecosystem, how can the public and private partnership strengthens our field, and specifically how might academic institutions like IIT Jodhpur is also doing, we can have collaborations done to build this ecosystem and have training programs in India specifically.
Thanks for that question, Shrishti. And I think it is paramount, So I think if you look at if you look at the global scale, if you look at US, which is very advanced right now, the whole advancement of science, or the whole application of science, or the whole translation of science occurs when there's a equal public-private partnerships. These are formal, informal ways of basically tying up. So as they say, see, the skill set first comes in the academic space, because they are basic scientists who are looking at science at a very deeper level. They have a functional understanding of the technology. They have a functional understanding of the application area. So it's very important that tomorrow, if we can forge ways of communication between industry and academia, and again, there are various ways. On a smaller scale, we can have small cohorts of meetings, to meet together and understanding what are the challenges which industry has, and which academic institute has expertise to solve that problem. And tomorrow, if an approach requires, like as I said, biology is complex. Diagnostic needs to get more deeper to basically stratify a patient for a personalized genomics approach. Today, people are using genomics approaches. Tomorrow, people will be using genome, epigenome, proteome, microbiome, exosomes, and the list can continue on. Now, as the data, I mean, as there are more layers of data which is coming out and the complexity is increasing, obviously the skill set in the industry requires hand holding by the academia to basically work together so that, as Dr. Mitali said, meaningful deductions are done. Otherwise, we might just probably make deductions based on maybe very isolated markers, which may or may not be very comprehensive, or we are just trying to copy paste Caucasian data into our context and basically making inferences. So to me, I think it's paramount that first of all, an open mindset between both academia and industry should be there to forge, partnerships. There are many ideas. I mean, I think I would love to see in India when a large scale government project has a both public and private component where, assuming the next large genomes, project, suppose India does 100,000 genome project, how industry and academia work together to complete that project, because the capacity is available at both the sides. So I think those kinds of initiative, if they can be done with all the checks and balances, put together for data security and stuff like that, that will, I think, pave way for a lot of industry academia collaborations.
Thank you, Praveen. Thank you, Professor Mitali, for answering and shedding lights on the importance of multi-omics and how we can establish an ecosystem and bring such collaborations further with other partners as well. So yeah, that's all from my side, Arushi. I think we can take questions from the participants.
Yes.